Hello, everybody. My name is Mukti Ryan, and I'm an instructor of anatomy and physiology at Spokane Community College. The textbook that I use for my class is Principles of Anatomy and Physiology by Jared Tortora and Brian Derrickson. I'm planning on recording a lecture series for anatomy and physiology, and the first three chapters have been clubbed into one lecture because most of it is a refresher on what you've already learned in your general biology class or Bio 160 classes. So one of the things I usually like to tell students with A&P is that repeat, repeat, repeat until you're bored and then repeat some more. So let's start off with talking about what anatomy is versus what physiology is. We're going to look at a lot of definitions. We're going to look at levels of organization, from atoms to molecules to macromolecules, that kind of stuff. Characteristics of life, which talks about properties that are exclusive to living things. And finally, a little bit more detail on the process of homeostasis than you've gone over in your general biology class. So talking about the study of anatomy, when you think of the term anatomy, it talks about study of form. So how something is structured comes under the study of anatomy. Now, structure can be studied using animals or using cadavers. So the best way to study human anatomy is by using a human cadaver. Comparative anatomy goes over the anatomical comparison between animals and human beings. It's a lot easier to get animal specimens than it is to get human cadavers. So, basic principle anatomy is a study of structure and the study of form. Now, in anatomy, we have quite a few subdivisions. Now, anything that you can touch, the term for touching is palpation. So any kind of anatomy that you can study using touch without cutting anything open comes under surface anatomy. So in order to study the structure of your facial region, you can use palpation. So that would be surface anatomy. Now, there are structures in the human body that you cannot visualize or study unless you make incisions and cut something open. So anytime you have to use dissection or cutting, that term is called gross anatomy. For example, in order to study the human brain, one has to get into the cranial cavity. So that is gross anatomy because you cannot study the structure of the brain unless you cut the skull open. The next subdivision of anatomy is developmental anatomy. So this talks about what happens during gestation and anything that is before 8 weeks of gestation is called embryology. So this is a study of how the embryo develops into the germ layers and what happens there. Now, we as human beings are extremely multicellular. So we're made up of millions and millions of cells, and cells of similar kind and that have similar function are known as tissues. So the study of tissues comes under histology. So when you take a tissue sample and you're looking at it under some kind of a microscope, that comes under the branch of histology. So for example, if a physician suspects cancer or a tumor, then a part or a section of that tissue is taken and sent to the histology lab in order to study it further. Now, the study of the structure of cells themselves is called cytology. So cytology deals with whatever's inside the cell. So we're talking about the different organelles, their functions, that kind of stuff. And finally, pathology is the study of disease. Now, when we say disease, we're talking about pathology is basically a branch that goes over a lot of different sub-branches. So it could involve histology, it could involve cytology, it could involve gross anatomy, it could involve surface anatomy. So pathology is a conglomeration of a lot of different sub-branches of anatomy. One thing to keep in mind for human anatomy and physiology is the concept of anatomical variation. Now, something is referred to as an anatomical variation if it looks different, but the function of the organ is not disturbed. So it functions normally, but it just looks different. That is when it is called an anatomical variation. So over here, what you're seeing, if I can use a pen, what you're looking at is the normal position of the human kidney. It's present on the dorsal side, so towards your back, and the abdominopelvic cavity. So you see the kidneys over here, this is a kidney and this is a kidney, and then you have the adrenal glands over here and over here. Now sometimes one kidney is far lower in the pelvic region and therefore is sort of detached or farther away from the adrenal gland. So this is an anatomical variation because the function of both the kidney and the and the adrenal gland is not affected. It just looks or is positioned differently. Another kind of an example for anatomical variation here is the horseshoe kidney that you see right here. The horseshoe kidney is when both the kidneys are fused into one. Again, function is the same, structure is what is different. The next concept is to study physiology. So when anatomy studies structure, physiology studies function. So how things work, how do tissues work, how do organs work, how do organ systems work, all of that comes under the study of physiology. We have, with every organ system, you have experimental science and you have a whole physiology division of that particular organ system. So endocrinology is the study of how endocrine glands function. Neurophysiology is how the nervous system, which includes the brain, spinal cord, and the nerves, function. Now, physiology is because a study of function a lot of times involves animal experimentation, which can become an issue when it comes to ethics. In pharmaceutical research and development, when you have a new drug which is being tested, it first has to go through in vitro testing, then it has to be tested on live animals, which in most cases are rodents or primates or livestock, cattle, that kind of stuff. And then finally, once it passes those tests, it goes through clinical trials, which is trial on consenting human beings themselves. So it does involve kind of gray areas about ethics. Some people are more comfortable with it than others. But the bottom line is that in order to study how something reacts, how something functions as a reaction to a substance, a drug, it is important to carry out those experimentation. Now, one of the basic principles that you probably have looked at during Bio 160 is that structure mirrors function. So what this means that things are structured a certain way because they have a function to fulfill. Now, our rib cage is structured like a cage because it has a function. So what do you think is the function of the rib cage? It protects our soft tissue organs, the lungs and the heart. So it's a cage that protects the heart and the lungs from impact. Another example is the bones of the skull. The bones of the skull are mostly compact bone, and they're very heavy. Now, they're structured that way. They're compact and they're heavy, and therefore harder to break because their primary function is to protect the brain. Another example is the lungs. Now, in the lungs, the job of the lungs is to, when we inhale, absorb oxygen and diffuse it into the bloodstream. So the air sacs, or the bronchi and the bronchioles in our lungs, are very thin so that there can be diffusion of oxygen from the air into the bloodstream. So if we had very thick air sacs in our lungs, it would be more difficult for the oxygen to be diffused. So structure mirrors function. Something is structured a certain way because it has a very specific function to fulfill. The next concept we're going to go over is the structural levels of organization. Now, right down at the very bottom, you have the chemical level. So under chemical level, you have the smallest units of matter, which is atoms. So if you've seen a periodic table of elements before, you know that hydrogen, carbon, oxygen, nitrogen, all of these are atoms. So within atoms, you have the nucleus, you have the protons, you have the electrons, and you have the neutrons, but an atom together, along with its nucleus and the electrons, is considered the basic unit of matter. Now, when you have atoms that combine or form some kind of a chemical bond with different atoms, it is called a molecule. So a molecule can be made up of two identical atoms, or it can be two different atoms. So hydrogen gas, which is H2, or oxygen gas, which is O2, is an example of two similar or identical atoms coming together to form a molecule. Now, an example of a molecule that is essential to life, which is water. Water is composed of two hydrogens covalently bonded with one oxygen. So atoms come together, form chemical bonds, and form molecules. Now you have different molecules that come together and form bonds such as hydrogen bonds and make bigger molecules. An example of that is carbohydrates. So carbohydrates is a very large molecule that consists of carbon atoms, hydrogen atoms, and oxygen atoms. So that is an example of a macromolecule. DNA, proteins, DNA is a nucleic acid, DNA, RNA. Proteins, lipids are other types of macromolecules. Then you move on to the cellular level. I'm actually going to flip over here to this image in order to explain it further. So what you see right here is the atoms. Atoms combine to make molecules and macromolecules. Here you see an example of the DNA molecule. Now, different macromolecules come together and form organelles. So under the cellular level, you have organelles. You have nucleus, you have the nucleolus, you have the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, all of that kind of stuff, mitochondria. Now, in order to have a fully functioning cell, you have to have all of these organelles. So organelles come together in an organized fashion and make a complete functioning cell. So if you remember the cell theory, you probably know that cells are considered the basic structural and functional units of life. So if you look at a cell, one cell in itself is able to fulfill the criteria for all of those properties of life that we're going to talk about in just a little bit. Now this is an example of a muscle cell. Now we have muscles that we can control at will. We have muscles that we cannot control at will. So this is an example of a smooth muscle, or a muscle that you cannot control at will, and hence is called an involuntary muscle. So different types of cells of similar type come together and serve a similar function, and that structure is called a tissue. So a tissue is nothing but a group of cells of similar structure and similar function that are connected in one way or the other. So what you see here at the tissue level is the smooth muscle tissue. So you have smooth muscle fibers that have come together to form a smooth muscle tissue. Now you have muscle tissue. Different types of tissues can come together and make an organ. So different examples of organs in our body is one example is the stomach. We have our intestines, we have the heart, which is an organ, the brain, which is an organ. So the stomach here is made up of smooth muscle cells. The heart is made up of cardiac muscle fibers. The brain is made up of nerve cells or neurons. So different types of cells come together with similar structure and similar function and form an organ. Now an organ cannot work by itself unless the supporting structures in that organ system are functional. And an example of that would be the systems level. So over here is the digestive system. So the fully functioning digestive system would have the stomach, it would have the large intestine, it would have the small intestine, the liver, the pancreas. So all of this comes under the organ system. So if you're thinking of, for example, if we were to think of the circulatory system, we would be talking the heart is the primary organ, but then you have the arteries which carry the oxygenated blood to all cells of the body, and then you have the veins that bring back deoxygenated blood to the heart. So that whole circuit is a fully functioning organ system. And for an organism, in this case us human beings, we need all of our systems to be fully functional. So different systems come together, serve basic purposes, and form a functioning human being. Whoops. Now moving on to the characteristics of life. So these are basically properties that something has to fulfill in order to be considered a life form. You've probably learned a little bit about this in your Bio 160 classes, and talked about the cell theory as well. So when you think of objects, when you look at this mouse that I'm using here right now, it is an inanimate object, but a real mouse is a form of life. So what is it that is different between a computer mouse versus a real mouse? The difference is that a real mouse is going to satisfy all of these 6 properties of life, whereas the computer mouse is not. So let's look at point number 1, metabolism. So one can define metabolism as converting one form of energy to another. So you could either build up or build down. So you could break down or build up. So converting one form of energy to another, exergonic and endergonic reactions. That is what metabolism is. When you're taking smaller pieces, process of dehydration synthesis is occurring and a bigger molecule is built. It is referred to as an anabolic process. When you have a bigger molecule that needs to be chopped up into its smaller units, the process of hydrolysis is taking place, and that is called catabolic reactions. So a real mouse can take one form of energy, change it into another. So it takes in oxygen, breathes out carbon dioxide. It's going to eat an insect and be able to digest that insect. It is going to be able to build muscle. It is going to be able to break down muscle as well. So that is an example of metabolism. The computer mouse not going to be able to do that. The next property of life is responsiveness. So human beings respond to their environment. The environment could refer to the external environment or the internal environment. So if you subject a real mouse to heat, it is going to want to move away from uncomfortable heat. If you subject it to intense cold, it is going to want to move away and get to a warmer place. A computer mouse not going to care about responsiveness. The third property is movement. And by movement, I'm not talking about moving the organism in general, but the movement of atoms and molecules and macromolecules within the cells and the organs themselves. So in human beings or forms of life, the movement that I'm talking about is osmosis, diffusion, the different types of diffusion, as well. Receptor-mediated diffusion, simple diffusion, that kind of stuff. So in a little bit we're going to talk about what osmosis and diffusion are, but this is movement of molecules within the cells and the organism itself. The next property, or the fifth property of life, is differentiation. So differentiation is what happens when two cells, the egg and the sperm, combine, form a zygote, and then 40 weeks later you have a human being. So that change from an unspecialized cell to a specialized cell is called differentiation. And an undifferentiated cell is usually called a stem cell. So a stem cell can differentiate or morph into a more specialized form. And the last property of life is growth and reproduction. So reproduction can refer to asexual reproduction or sexual reproduction. Now the cells in our body reproduce by mitosis and meiosis. So making more of yourself, making copies of cells or the organism itself, is called reproduction. Now when you talk about life, you cannot talk about it unless you talk about death. So a human being, a lot of times in movies and TV shows, you say that somebody XYZ was pronounced dead. Now there's legal death and there's biological death. They're two different concepts. Now somebody is considered legally dead when they have no brain waves for 24 hours. Now what are brain waves? The brain is made up of specialized cells called neurons. And the neurons basically transmit tiny electrical signals. So they communicate. One neuron can communicate with another neuron via a very tiny electrical signal. So if a person's brain is not generating or transmitting these electrical signals for 24 hours continuously, then they are considered brain dead. The person is also considered legally dead if they have no reflexes. What is a reflex? A reflex is a movement of your muscle or some kind of a reaction which is involuntary. An example of a reflex is when you touch a hot surface, you reflexively pull your hand away. If someone shines a light in your eye, you can see your pupil contract or become smaller. So that is a reflex. So if someone doesn't have brain waves, or if their brain cells are not communicating with each other for 24 hours, then they're not going to have any reflexes. Another criteria for legal death is when the person is hooked up to a heart-lung machine. So their heart isn't beating by itself unless an external source is giving the heart that electrical input that it needs to continue to beat. Now this was legal death. Now this person could be legally dead, but that doesn't mean that every cell in their body is dead. So biological death is when each and every single cell of the body is dead. So this takes a few hours after a person is pronounced legally dead. And this is one of the reasons why when someone is deceased, their organs can be used for transplantation. So this is an example why someone can donate their eyes after death, because the cells in the eye are able to stay alive for a little bit longer than other cells in the body. So you have cellular or biological death, and you have legal death. The next concept that we're going to go over is the concept of homeostasis. So everybody knows by this point that homeostasis refers to a steady or stable internal state. So the human body maintains a normal range of a lot of substance. Temperature is regulated to be in a normal range. The amount of insulin that you have and the amount of blood sugar that you have, normal range. Thyroid hormone, everything has a normal range. And our bodies have the ability to maintain these numbers in their normal range, irrespective of what the environment might be. So that is called homeostasis, and it's usually regulated. So a lot of different systems have to work in conjunction with each other in order to maintain homeostasis. And it usually involves body fluids. So let's look at the different body fluids that are involved in homeostasis. They can be broadly classified into two categories. You have the fluids that are present inside of a cell, which are called intracellular fluids, and then you have fluids that are located outside of the cell, so in the extracellular space. Hence, they're called extracellular fluids. Some of the fluids, one of the most important bodily fluid is blood. So what blood is water, dissolved gases, clotting factors, proteins, enzymes, and we have blood cells floating around in the blood as well. So blood plasma is an extracellular fluid within blood vessels. So I'm going to take a minute to explain what blood versus blood plasma versus serum means. So blood consists of clotting factors, dissolved gases, and blood cells. Now, blood minus white blood cells and red blood cells is plasma. Now, if you take plasma and if you remove the clotting factors out of plasma, CF stands for clotting factors, you end up with serum. So make sure that you can differentiate between blood, whole blood versus blood plasma versus blood serum. Lymph is another extracellular fluid. So lymph is the extracellular fluid of the lymphatic system. It involves the thymus, the lymph nodes, the spleen, and the lymph fluid itself. So lymph consists of white blood cells and is kind of like the drain-off mechanism. It's part of our immune system. Cerebrospinal fluid is an extracellular fluid that surrounds the brain and spinal cord. So this is a sterile extracellular fluid. It's flowing in an enclosed cavity around the brain and the spinal cord. The next couple of extracellular fluids are synovial fluid, which exists between the major joints in the body, the larger movable joints in the body, for example, your elbow, your knuckles, your knees. They're filled with a fluid called the synovial fluid, which provides nourishment, it provides shock absorption, kind of keeps the bones healthy. Aqueous humor is the gelatinous fluid, which is the extracellular fluid in your eye cavity. So the eyeball is hollow on the inside and is filled up with aqueous humor. Now, when does homeostasis change? Or homeostasis is constantly being challenged by whatever's happening in your external environment. So the external temperature could be more than your normal body temperature, or it could be less than your normal body temperature. It could be an environment where there is a lack of oxygen, there is a lack of food. So that comes under physical insults. So stuff in the environment, the external environment, could cause a change in your homeostasis. Changes in the internal environment. So your blood sugar drops when you haven't had anything to eat. So that is an example of internal environment being challenged. And then you have physiological stress. Stress can cause the disruption of homeostasis when it comes to hormones or just chemicals in your body. Now, when we talk about homeostasis, we're going to talk about feedback systems. Now this image, I like to explain this image because anytime you have homeostasis, you have to have a complete circuit. So the complete circuit consists of the first key player in the circuit has to be a receptor. So a receptor is a structure of some kind. It could be present inside your body or it could be present on the outer surface of your body. So a receptor is something that senses a change. So for example, we have something called thermoreceptors in our skin that detect temperature difference. So we know that it is hot outside because there are thermoreceptors in our skin that are detecting that change. Now here's what I'm going to introduce two new terms to you guys. An afferent signal versus an efferent signal. One starts with an A, one starts with an E. So anytime, now when the receptor has detected this change, the next step is going to be for this receptor to send a notification to the control center. And in the human body, the control center is typically part of the nervous system. So it's either located in the brain or it's located in the spinal cord. Now, anytime you have a signal that is going inwards towards the nervous system, it is called an afferent signal. So a signal coming in towards the spinal cord or towards the brain is called an afferent signal. So the thermoreceptor sends an afferent signal to the control center, which is part of your nervous system, and the nervous system is going to send out a signal of its own. So an outgoing signal from the brain or the spinal cord is called an efferent signal. And the efferent signal is going to go to some kind of an organ. Now in this temperature example, if you touch a hot surface, if you accidentally end up touching your finger to the stove, the thermoreceptor is going to sense that that's too hot, and afferent signal is going to go to your nervous system, to your central nervous system, which is either the brain or the spinal cord. In this case, it would be the spinal cord. And then the spinal cord would generate a signal of its own called the efferent signal. And where would this signal go? So in order to be able to remove or take back my hand from the hot surface, I'm going to have to use my skeletal muscles. So in this case, that efferent signal is going to an organ, which is my skeletal muscle. So this organ is called the effector organ. So can everyone see how this is a closed circuit? First, something happens, homeostasis is disrupted. That disruption has to be sensed by a receptor. Once the receptor senses it, it generates an afferent signal, which gets carried from your nerves to your central nervous system, which is the brain or the spinal cord. The brain or the spinal cord is going to generate a signal of its own called the efferent signal, and the efferent signal is going to be sent to the effector organ in order for homeostasis to be restored. So let's look at the example of, there are two feedback loops. One is the negative feedback loop and one is a positive feedback loop. So the negative feedback loop is when there has been a change, that change is not good, and that change has to be reversed. So reverse is negative. So reversing a change is what the negative feedback loop is. So this is an interesting example. Blood pressure regulation. So blood pressure is basically dependent on how wide or how narrow your blood vessels are, and how fast your heart is beating. So the faster the heart rate, the higher the blood pressure. The slower the heart rate, the lower the blood pressure. So people's blood pressure is going to be higher after intense exercise than if they're just sitting on the couch watching a comedy show or something. But the receptor in this case is called a baroreceptor. because anything that has to do with pressure is baro. So a barometer detects atmospheric pressure. So a baroreceptor is a pressure receptor which is located inside our blood vessels. So blood vessels are what carry blood from the heart to the tissues and from the tissues back to the heart. So they have baroreceptors. Something happens and your heart starts to beat faster. There has been a disruption. So normal blood pressure is about 120/80. So something happens and your heart starts to beat faster, your blood pressure goes up. The baroreceptors are going to detect this change and they are going to send an afferent signal to the brain, which is the control center. The brain right here is going to interpret that change, make a decision of what needs to be done, and it is going to send out an efferent signal. So what do you think in this case is going to be the effector organ? So the only way to reduce blood pressure is to slow down the heart rate. So the efferent signal is going to go to the heart, which is the effector organ in this case, and the heart is going to slow down. When the heart slows down, blood pressure decreases and returns to normal. So can everyone understand how, in this case, there has been a change, and when the body reverses that change, that is called the negative feedback loop. Another example of the negative feedback loop is thermoregulation. So thermoregulation refers to our body being able to maintain an internal body temperature. So 98.6 degrees F is the normal body temperature. And no matter how hot it is outside, no matter how cold it is, our body maintains that temperature, unless you have a fever, and that's something totally different. But in normal circumstances, body temperature being maintained around 98.6 degrees F is another example of homeostasis. So in this case, first let's look at what happens when the environment is extremely hot, or your body temperature for some reason goes up. Now everyone knows what happens when your body temperature goes up and your body needs to cool off. You end up sweating. So let's see how that happens. So thermoregulation, the receptors in this case are thermoreceptors. So you have receptors in your skin, you have receptors in your blood vessels that are sensing the temperature of your blood and your skin. Now, there's been a disruption if for some reason your body temperature has gone up. So in this example, this woman's body temperature has gone up to 99.5, which is above the normal range. Now the only way her body can cool off is by sweating. So the thermoreceptors are going to send an afferent signal to the brain, and the brain is going to generate an efferent signal, and it is going to send it to an efferent organ, or an effector organ. And in this case, the effector organs are going to be the blood vessels. Now, when the effector signal, or when the efferent signal goes to the blood vessels end up increasing in diameter. When the blood vessels increase in diameter, that is referred to as vasodilation. So the blood vessels end up dilating, and therefore that causes a person to sweat. And the person is going to sweat to cool off until their body temperature returns to around 98.6 F. Now let's look at the opposite process. So for some reason, the person's body temperature has dropped to 97.7 degrees F. So when a person is cold, and a person can warm up by shivering. So shivering basically generates heat so that the body temperature can come back up to normal. So thermoreceptors detect a change, afferent signal goes to the brain generates the efferent signal, and in this case, the effector organ is going to be two things. One, the blood vessels. The blood vessels are going to constrict. That is called vasoconstriction. And when there's vasoconstriction, there's also an efferent signal going to the skeletal muscles to rapidly contract and relax. So when the muscles rapidly contract and relax, that process is called shivering. So that generates a byproduct of that is heat. The next concept is a positive feedback loop. So positive feedback loop is quite different from a negative feedback loop because the positive feedback loop involves creating a change. So a change is sensed and a change is amplified before it can go back to normal. One of the best examples of that is the process of childbirth. So here's what happens during childbirth. So here's a fetus in this woman's uterus, and typically close to labor, the head of the fetus is creating pressure or pushing against the woman's cervix. So when that pressure, when the pressure becomes intense enough, the receptors in the cervix are going to send an afferent signal to the brain. Now in this case, the brain is going to create an efferent signal, and it is going to cause the pituitary gland to secrete oxytocin. So oxytocin is a hormone that ends up causing uterine contractions. So if anyone's ever been induced using Pitocin, that's nothing but synthetic or artificial oxytocin, which your body naturally produces. So in this case, oxytocin is going to generate, or it's going to create a change. It's going to amplify something. In this case, it's going to amplify the uterine contractions. The more the oxytocin, the stronger the uterine contractions. So the uterine contractions get stronger and stronger and stronger, therefore causing the fetus to be pushed out of the cervix through the birth canal, and that is when the woman ends up giving birth. So with this lecture, I'm going to stop here today, and with the next one, I'm going to continue with the chemical level of organization. Okay.